Good afternoon. It's Saturday at EHA 2018. I'm Blake Morrison with World Myeloma Forum. I'm here with Paul Richardson. Dr. Paul Richardson from the Dana-Farber Cancer Institute, world renowned for myeloma research and treats a lot of patients.
Yeah.
He's here to talk to us about a really interesting trial called OPTIMISMM. It's a triplet versus doublet therapy in Len refractory disease. So why don't you tell us a little bit about the study?
Great, thank you Blake, and good afternoon everyone. It's an absolute pleasure to be here. Blake, I think the essence of the OPTIMISMM trial is summarized by the fact that lenalidomide-based therapy is being used up front now as a standard of care, certainly in the United States and increasingly elsewhere. And lenalidomide is now being used as part of maintenance, both in transplant eligible and transplant non-eligible patients. So the goal of OPTIMISMM was really to establish a real-world platform that could address this area of relative unmet medical need. In other words, when lenalidomide fails, what do we do? So we built the platform on the premise that obviously after IMiD runs out of benefit, a proteasome inhibitor-based approach makes sense. And obviously we had great data from the synergy of IMiDs and proteasome inhibition from the original VTD, RVD, KRD, IRD concepts. And combining POM with Bortezomib had been shown in our phase 1/2 experience to be safe, well tolerated, and very importantly, very active. We also knew that you could combine POM successfully with carfilzomib, you could combine it with Dexamethasone, but obviously we wanted to provide a real world platform. So the idea was to take forward a triplet versus a doublet in Lenalidomide-Exposed and Lenalidomide-refractory patients. 1 to 3 prior lines, and this was a global effort, really a tremendous international team. And essentially what we also built into the design was that patients continued on therapy, so it reflected the paradigm of continuous treatment. So there was no fixed duration of therapy. People got pomalidomide, bortezomib, and dex for up to their 8 to 9 cycles, similarly for bortezomib dex up to 8 to 9 cycles, and then at that inflection point converted to maintenance strategies in both arms, which I think was the strength of the trial, because obviously a lot of recent phase 3s have, one, been in patients in whom Len exposure has been minimal, and two, there's been fixed duration to the control arms at least, which has led to some difficulty in exactly interpreting the impact in the clinic of what the comparisons might mean. Sure. So in terms of results, we enrolled approximately, actually 570 patients, approximately 566 to be exact, and we were very pleased to see a very equal distribution in characteristics, the beauty of a randomized trial. 100% were Len exposed. 70% of the patients were truly Len refractory, which is important. We did allow prior Bortezomib exposure, but they could not be refractory to therapeutic Bortezomib for obvious reasons, because then the equipoise of the control arm wouldn't apply. So with that in mind, we did our analysis for the primary endpoint. We looked early, Blake, because we'd had data from studies like CASTOR, which showed that the PFS for Bortezomib dex was around 7 months, and therefore we, by protocol amendment, did an interim look earlier than originally planned. So with an early look, which obviously has its risks, we were very pleased to see that the PFS benefit was significant and clinically meaningful. Basically a 4-month difference in favor of the triplet, with a PFS of approximately 11 months versus 7 months for the control group. What was particularly striking, though, was if you looked at the first relapse population. If you looked at those patients in first relapse, the PFS benefit was 20 months versus 10 months. And that group of patients in particular had about a 10-month difference in their favor, which is very important. And the clinical question that's arisen is, what about the Len refractory patients in that first line? And we were very pleased to see that the PFS for the control group was 9 months, but actually for the triplet it was 17.8, with a hazard ratio of 0.55. So, really exciting data that in first relapse, not only can you improve clinical benefit by a solid 10 months in everyone, but even if you're Len refractory, you get a significant clinical gain. And so if you look at response rates as obviously a surrogate for this kind of clinical benefit, 80% overall for PVD versus 50% overall for VD in first relapse, 90% for PVD, 60% for VD. And I think what's really nice about that, Blake, is it sort of confirms the synergy that you and I have known about for a long time between IMiD and proteasome inhibitors, and at the same time in a very manageable, practical context.
So two questions. In terms of it establishes also IMID to IMID,
Exactly,
right?
exactly,
Activity and being able to sequence within the class.
Yep.
Question about tolerability though,
Yep.
was there major differences between the two arms in terms of patients tolerating the triplet versus doublet?
Great question. That was no unanticipated safety signals.
Okay.
What we saw was basically the duration of therapy was much longer, as you'd expect for PVD, so therefore obviously you have to take that into context when you look at the aggregate toxicity data. But suffice to say, neutropenia was probably the biggest difference, but it was manageable. Our rates of febrile neutropenia were just 3%, so that reflects that. Deep vein thrombosis, very low rates, but there with the triplet, not with VD. But again, very low rates and manageable. Neuropathy, a little higher for PVD than VD, but again, not huge. And so in general, you looked at a very comparable safety profile for both groups. And then when we looked at subgroups, what was really interesting, for patients with high-risk cytogenetics, renal dysfunction, older age, the benefits were consistently good across all groups, which is very important, obviously, from a practical point of view.
Is MRD testing being done in this patient line of therapy?
It is, and MRD testing is cooking, so is other correlative science.
Okay. And I would presume you're going to look at overall survival as well as
ultimately, yes,
endpoint in this study.
Yeah, as a secondary endpoint, yes, but obviously that's going to, thank goodness, for the benefit of patients, that's going to be a long time.
Yes, I was going to say it'll be interesting to watch this study continue to mature and generate interesting data. But thank you very much for sharing what you did today, and we really appreciate you stopping by.
It's my pleasure, Blake, and thank you.
Thanks
very much.